Okay, so it's a great pleasure to welcome my friend and long-term collaborator, Kristin Swanson, and of course, co-chair of this exciting enterprise. I guess it's probably worth sharing a little couple of anecdotes just so you sort of understand where we're coming from. So, but you probably didn't know that Kristin Swanson is actually Jim Murray's last PhD student, and she got her PhD with him in '99. And if you don't know who Jim Murray is, then you don't know the field of mathematical biology. She's one of the first people that I ever met that was already working with clinical data. And she's been doing that for over 3 decades. So, makes us, the rest of us feel a little bit embarrassing. Yes. And we've been, I guess I've known Kristin for maybe on the order of 2.5 decades. But she's really a pioneer. And it's something that I think hopefully you'll see in this work, but it's just worth putting it in context that she's always been ahead of the curve. So Kristin.
So I wanted to talk about some didactics, moral truths to share today. So I thought some theme that would resonate with this group would be some of the work that we've been doing and highlighting and focusing on some of the work that we've been doing on the interface between machine learning and sort of integration of mechanistic models. So I'm actually going to give a background of the type of work that I do and the type of modeling that I've done on the mechanistic modeling side, and then show how we're kind of infusing that into machine learning approaches to advance patient care in brain cancer patients, and ultimately detect modeling and detecting shifts in the patient's tumor ecosystem in vivo. So we all know the challenge of really every cancer, but the case of glioblastoma is particularly profound. Every patient really does deserve their own equation because every patient responds so differently to any given treatment. So this is a cohort of 15 newly diagnosed patients with glioblastoma, and they're all going to receive the same treatment. They're going to all receive the standard of care, radiation and concurrent chemotherapy, temozolomide chemotherapy, followed by adjuvant chemotherapy. And there's some patients for which their tumor just blows right through the growth kinetic. And then there's others that are actually very well controlled on this disease. And the slow, and I mean like many decade progress that we've had, which is incremental at best, has been these long tail patients mostly that have contributed to this shift in the dynamic. So in the current case where the dominant chemotherapy in this situation is temozolomide chemotherapy, this tail has had kind of an increasing abundance in the population of patients relative to previous times. Well, what's true is that every patient is very different across this, and we would really love to have as many tools in our arsenal. If you're a clinician and you're a patient facing this sort of map ahead of you for the same diagnosis, the same histology under the microscope, you really want as many tools as possible to say, at any one time point, can I say, course, is the treatment actually making this go down? Because it turns out it's not that simple. And second, can I predict ahead of time which patients are going to be which? So I'll tell you kind of a couple of vignettes about both aspects of that. So just reiterating the dim progress within this disease, that we're still sort of at a median survival of something like 14 to 16 months. So my work started very traditionally under Jim Murray's approach of using lots of reaction diffusion equations. So my entire world, my entire training was really about partial differential equations, reaction diffusion, pattern formation, things like that was really the sort of mathematical background that I came to this with. So when I was very interested in working on cancer, my father had just died a couple months before this of lung cancer, and I was focused in cancer, very happy to not work on lung cancer. But it met up with a very wonderful neuropathologist who became a very close friend and mentor shortly after meeting Jim, who introduced me to him. His name is Buster Alvord, and his spirit is really intrinsic in all of this. What's notable about this case of, and what really drew my interest in the space of glioma is the fact that it's such a complex disease because it's tumor cells that are diffusely invading into the normal brain and really interacting in that normal brain environment. So the first thing you know about this disease is this movie right here, where you're seeing diffuse invasion into the black normal brain parenchyma, where there's just, normal brain cells and regular, cells out in the periphery, and only the tumor cells are labeled in red. And you see they're moving very large distances, even over a small, in this case, 24-hour window. So as a mathematician, I can look at this and go, that's a complex system of interacting agents. Let me see if I can figure out some rules for that. I can track the individual cells. Turns out they do a diffusion process pretty nicely. I can track the proliferation, kind of, get a doubling time. I can write down equations like that, reaction diffusion equations, that tell us about the rate of change of the local glioma cell density as a function of this dispersal that we're seeing on the screen, and the proliferation that's happening in these tumors, and hopefully response to whatever treatment where we want to model or is actually being applied to these patients. So we have a long history of being able to take this sort of tumor forecasting model technique, applying it and personalizing it to the patient's MRIs, and allowing us to get ancient movies, this movie is over 20 years old, of what the MRIs evolving over time as distinct from what the actual diffuse extent of the tumor is, as a result of that diffuse invasion you saw with those red individual cells. So we have a long history of being able to tune these models to patient-specific parameters, and then use that to sort of advance our cause of understanding preemptively what treatment might do for a given patient. If one patient has a lot more diffuse invasion, that actually matters a lot to surgical resection. It turns out if you have a larger penumbra, patients do much worse, even after gross total resection of the imageable abnormality. So really, imaging this will be a theme. Imaging only tells us the tip of the iceberg of the disease, and it's even less clear than that. It's more like the reflection of the views of the physics through a lens of what MRI is actually doing. So taking techniques like this, we've built frameworks that allowed us to take patient MRIs, segment out the abnormality in red, simulate a temporal lobectomy in this situation, overlay the radiation fields that we extract from the radiation treatment facilities, and be able to build these sort of predictive models. And I think somebody pointed out, Russ, this is central to his work when he was doing his PhD in my lab, where he was building these predictive models of the radiation sensitivity in each of these individual patients. So we can do a lot in this space, but I'm actually not going to talk about that today. What I will talk about is the need for validating models like this is collecting a ton of patient data. So one of the things that we've done to validate sort of our suite of modeling approaches and develop our suite of modeling approaches has been to continually collect and recruit patients into a large sort of repository, which we call a patient view database. It is accessible by collaboration, so come talk to me. You're welcome to join in. We currently have over 5,000 patients in it with over 50,000 MRIs that are already segmented. And what that means is I've got the temporal evolution of any given patient's tumor relative to whatever treatments that patient is receiving on the bottom of that, x-axis there. And so there's a rich data set to be able to validate that. So when I moved to Mayo Clinic about a decade ago, a little over a decade ago, I decided, well, okay, this sort of work has been really informing us, but one of the biggest challenges we were facing was the disconnect between the biology that we saw in the microscope, under the microscope, versus what we could see on the patient's MRI. So at Mayo Clinic, I started this image localized biopsy protocol that allowed us to collect MRI localized biopsies during the course of a patient's treatment. So here's a typical patient. He's a 58-year-old male with a grade 4 astrocytoma. And what you'll see is a couple annotations here. In cyan is the outline of a T2 weighted MRI. In red is that same red that I've been and the extent of the T1 GAD MRI, and in yellow are the biopsies, where they're locally collected within this patient. And this patient just happened to come back 5 months later, had an early progression, and had another set of biopsies. So being able to capture and quantify the diversity of both the spatial landscape and the cellular landscape relative to that spatial extent has been an important thing to now then map to the local regional MRI features to better understand what biology is MRI revealing to us or not. So to summarize that, it's a complex course of working with the neuronavigational systems in the OR. There's literally wands that are used in the surgical resections that are basically have GPSs on them. And then the GPS reflects on the wall and tells you where in the MRI space you are, the XYZ coordinates, and you basically record that information. And there's some planning working ahead of time with the surgeons, but there's a lot of manual effort required to collect these sort of samples. But once we collected these sort of samples, we now have a new cohort of almost 500 new samples that we've just recently completed analysis on. So there's lots more to come in this space. But everything that I'm going to talk to you about today was really focused on those first couple hundred MRIs, localized biopsies, where we're getting about 5 to 10 per patient. Okay. So now using this data, we can go back to what we do know about the complexity of our microenvironment. Well, we know tons of things about how the MRI physics reflects on the tumor microenvironment, what it thinks it says. So you can line up these biopsies to not only the routine sequences, T1 GAD, T2 MRI, but also the advanced MRI sequences that are further down that we've heard a little bit about. Now, then, at each of these image localized biopsy locations, we can collect histology, transcriptomics, genomics, you name it, whatever we might be interested in learning about, whatever might be. And then they have the local regional imaging features. And that is a classic machine learning problem, right? You have this high dimensional pattern of imaging features. You have a low dimensional answer, picture, histology, tissue state A. There are 3 of those kind of cells there, whatever you want to learn on. So you can build lots of different AI-based machine learning sort of approaches to build a map that now connects the MRI local regional features to the biology, even though we have limited samples. And that allows us to fill in not only where the biopsies were, we can put that on the map, but we can also fill in the blanks in between and talk about the ecosystem throughout. And that allows us an in vivo view of the biology as it's evolving over time. So one of the first examples in the theme of bringing together this proliferation invasion model, the model I showed you before with the temporal evolution of the reaction to fusion with the tumor cells highly invasive, taking that model and then combining it with this sort of machine learning approach where you could take these local regional imaging information and say, can I tell from the imaging patterns alone how to predict where tumor cells are? Let's just start with that simple question. And that's what this paper did. So we took these MRI localized biopsies, we lined them up to the MRI, we looked at their histology and got the answer. And the answer was how many cells were there, how many tumor cells were present. We then took the multi-parametric MRIs and got the additional features that are from all these different MRI sequences that you have. And we then, for each patient, generated the proliferation invasion model, the PI model, the model I showed you in the previous slides, and overlaid that as a map as well. And the net result is we could then train a machine learning model that was trained on not only the limited labeled biopsies samples that we had collected. There's some unlabeled samples because we know like far out, there's no tumor here, so we can use those as controls. And then add in whether we want to see what the benefit is of adding in the proliferation invasion model to that model learning. And it turns out you get very different pictures. So if you look at the machine learning models, they're kind of bumpy and they're kind of heterogeneous and they're like predicting, like in this bottom right picture, like they're predicting tumor cells, high tumor cells way out at the edge of the abnormality, which was just kind of odd, not really as logical as one might expect. Whereas the PI model does exactly the opposite. They're like, it's a gradient, it's a reaction to fusion, so it's going to be just like this nice little smoothish gradient of things. Whereas what's interesting here is the combined MLPI adds a lot of the textural features that you get from the machine learning part, the kind of bumpiness of things, whereas it adds basically a smoothness that is generated from the PI approach, proliferation invasion approach. So when we compared these different models, we literally just asked for these unseen biopsies, was the machine learning model better, was the PI model better, and was the machine learning combined with PI model better? And there's a couple additional details I'll share in just a moment. We found that the accuracy went way up when we focused on these top 2 lines of the accuracy of the combined MLPI model. And specifically when we did the patient-specific MLPI model, where we took any information that was specific to that particular patient at the time of surgery, the accuracy went crazy way high. But all of them were higher when we went in this direction. And specifically, here's what that looks like. So if you just took machine learning approaches at all, in the leftmost column, you'd look at all samples, and all samples are all the ones in the core of the tumor and the samples that are, collected from all kind of the edge as the surgeon is approaching the tumor. So what we call the brain around tumor, BAT. And then you see on the left, there's a decent correlation between the machine learning model and the actual pathologically determined tumor cell density. There's a between the PI model and the pathologically determined tumor cell density, but it's looking really pretty at that patient-specific MLPI model. Like the regression on that looks really good for the prediction of the model versus the actual data. So these are unseen samples. And if you limit those to only those samples that are sort of at the periphery, which is really where the question is, it's not really a question, is there tumor where I know there's tumor? It's, I know there's some abundance of tumor out here, but I'd really like to know how much is invaded out here is the real question. The brain around tumor samples are the really important ones. And you see that regression is really quite beautiful. Additionally, when you ask the modeling approach, the machine learning, what inputs were the most important in terms of it coming up with its answer, you find that the PI model is the number one thing that it found useful. So there's this huge complementary opportunity to combine all this mechanistic insight, which we have from exceedingly simple models like the PI model, but informing that into our machine learning space is highly logical, I think. Otherwise, in this data set, we certainly do not have sufficient data to do a full machine learning approach. There's just probably never enough data to do a fully machine learning approach and expect to be able to predict everything you'd want to predict. One, because it's hard to collect these samples. And two, you would need the diversity across patients are extremely wide. So it's like heartening that the machine learning is doing some pattern searching, but the baseline patterning referenced by the PI model really boosts the accuracy in ways that are really, really impressive. So everything I've told you so far has really been focused exclusively on the idea of invasion and peripheral invasion into the brain around tumor, things like that. But the biology of this disease is much more complex. And since we have these cool image localized biopsies, we can really look at that complexity at a much higher dimensional perspective. So we could look at this phylogenetically. So we're working some work with Noemi on this. We can try and connect the dots on our understanding of the genomics of these patients and the diversity across the regional atlas of these tumors. But we can also look at the cellular composition within these samples. So we do this cellular composition deconvolution using bulk RNA-seq with a single cell signature that we've developed ourselves from a large cohort of patients where we have single nucleus RNA-seq. And what we've found is that even biopsies that are very close to each other, that you'd be like, okay, well, nearness matters, should matter-ish, like some, maybe, I don't know. Turns out doesn't matter all that much. So if you look at these maps up here, even biopsies that are quite close to each other don't necessarily correlate all that well with each unit on this. This axis here is the relative abundance of different cell populations. You don't have to really read it from the back of the room, but like the top line is how many neurons are there. Next line is how many astrocytes, like boring astrocytes, reactive astrocytes are there, and how many oligodendrocytes. So this is like normal brain stuff on the top. And what's interesting is lots of normal brain stuff is quite enriched in these samples that are right at the edge of the tumor in 1 and 2. Right, but there's also cases where neurons showing up in other locations. And towards the bottom, you're seeing lots of T cells and immune reactive tumor-associated macrophages and monocytes. So the details of the cell populations aren't that important. What's important is those columns don't match each other, and if it doesn't really matter. The correlation between any compositional pair-wise of these is all over the place. It's everything from literally 0 correlation to 1. Next. So the predictive accuracy is in need. Like our understanding of what that cellular ecosystem is just not at all wrangled. So like we can talk about tumor cells being invaded, but the tumor ecosystem and who's present in what location is a vast unknown. But what's not unknown is the vast amount of MRI data that we have and the limited amount of image localized biopsy data that we have. So taking this sort of data, we have a large E54 that's focused on Center Grant, that's focused on how do you simplify this complexity down a little bit, the high cellular ecosystem diversity, into some sort of cohesive range that is tenable. So the way we approached this was to look at some single cell data. So this is single nucleus RNA-seq data. And we looked at basically the relative abundance of each in any given sample of whatever of the populations that were present. And these are the 18 cell populations. Now, 6 of those cell populations are gliomas, the GL underscores. And the other ones are either immune cells, reactive cells, or normal brain cells like oligodendrocytes or neurons. And even if you just do a simple PCA, you basically find 3 corners. You've got one corner where the neurons want to hang out with other things that look like neurons, like to be around neurons, like oligodendroglial progenitor cells. They like to be near neurons. Unreactive astrocytes. They hang out. So that's a region which you would have diffusely invaded tumor cells and mostly normal brain. In the bottom left, you have all the CNV positive samples where there's tons of tumor cells present and they're proliferating. They're actively growing. And in the top, you kind of have this immune inflamed inflammatory cohort of a community of cells. There's tumor cells in there, but there's a really dominant like immune inflammatory response. And so in the world of looking at immunotherapies, it's really important because it turns out immunotherapies are pushing you towards that state. And we are used to measuring the change in the proliferative state. Like, we think of cytotoxic therapies as just seeing changes in this. But the shifts towards this immune inflammatory is just confusing from an MRI perspective. It's not simple. It is not solved. And we're hoping to contribute to solving that. So when you take that sort of data and then do some spatial transcriptomics, looking at these single cell states, so here's my annotation of the different single cell states of the glioma cell states versus the non-neoplastic states and their substates, meaning astrocytes are not just one pile. There's at least three different piles, three different phenotypically distinct piles. The myeloid states, the tumor-associated microglia, macrophages, and the T cells all have their own pile of states. When you look at it in a spatial transcriptomic sense, you see that same clustering. There are CHIs, there are communities of cells that want to hang out together, and there's kind of these three clusters that kind of organically appear. And so I feel, as a mathematician, much more comfortable dealing with three things than the 18 bazillion vector of all things. So this is kind of the rubric that we're using to condense some of this complexity. So in short, this is what that rubric looks like. In the bottom left corner, we have this invaded tumor and mostly neural tissue, normal brain, and then it's got some diffusely invaded tumor cells. In the top is that inflamed reactive gliosis, reactive astrocytes, and mesenchymal tumor cells. And then down in the bottom corner, we have much more of those proliferative tumor cells surrounded by a very proliferative positive niche for those cells. Now, we can go back to all of our image localized biopsies and do some simple pseudo-time approaches where we can take this very high dimensional complex RNA-seq data and project it down to a lower dimensional space. And miraculously enough, I schematized it here, but it turns out there's three arms that you can get down to, which are representative of these same three arms. So my point is this triangle is going to serve the rest of our presentation about connecting sort of that more immune inflamed, proliferative versus diffuse. So this is 7 samples from a single patient, and you can see they're kind of covering all of the space. They don't live in one little niche. They're like everywhere in the space. And importantly, when you introduce treatment, they evolve, they move. So in this example, this is just a vignette of an example, looking at these 3 states in a pre and post-treatment setting for local convection enhanced delivery of topotecan. What you find is there is a shift in these tissue states as a function of treatment. And those sorts of shifts are the ones that we would love to look at clinically, right? So building models that not only tell you where are tumor cells, but tell me what state is the overall tissue system in would be extremely useful. So that brings us back to our high dimensional MRI sequences that all have sort of different colloquial understandings of what they mean. We do know that gadolinium, the brightness you see on T1 weighted MRI, does reflect blood-brain barrier disruption, but so does the vasogenic edema. It's secondary to this blood-brain barrier disruption that's represented on these T2 weighted MRIs. The perfusion MRIs, RCBV, is going to tell you something about vessel volume, angiogenesis. They're all associative. And it turns out they're not that very good at their associations actually being true, at least in our hands. So for example, if you take this practical utility of most basic MRI any patient will get, they go to the emergency room, this is the type of MRI you would get. You get a gadolinium enhanced T1 weighted MRI. You look at the margin of the core, you can see there's lots of different cellular heterogeneity, whether you biopsy in those locations, and you do see that contrast enhancing, meaning the brightness on the MRI does reflect more tumor cells. So this is the tumor cell density reflected from our image localized biopsies versus the non-enhancing. You can see that there's a difference, but there's really low specificity and sensitivity for that. It's lots of overlap, right? So that's not great. And this simple T1 GAD MRI can't tell you the difference between the immune arm, the diffuse arm, and the proliferative arm. So you're just lost. So clinically, this means yeah, we think there's more tumor where it's bright, But I couldn't tell you if my immunotherapy is doing great and they're all orange, or if all the samples that are in those regions are all orange, or if all those samples in that region are highly proliferative and the tumor is just blowing through. We can't differentiate the basic biology that we think we can using routine MRI. So then go back to these same MRIs and let's get some advanced MRI. We'll do the perfusion next. So now picking on perfusion MRI is often used in the recurrent setting in the case of glioblastoma, and it is classically used to differentiate regions of tumor-rich abnormality. So such that surgeons are like, if the RCBV is high in the recurrent setting, they'll be like, okay, we're worried there's tumor there, so we're going to go in and we're going to get that. And when they do, there's no difference. Perfusion is not telling you where there's tumor cells. There's no difference between those two distributions. And also it fails to reflect the cellular abundance, the cellular ecosystem, the overall tissue state as well. So the point is the routine tools that we use in the clinic aren't actually very good. And that's not at all surprising because if you're in the clinic one day, you can find that out real fast. So this is my last 3 slides, which is basically then taking that same sort of approach of putting in a very simple, in this case, an extremely simple model into a deep learning framework with those same sort of biopsies to be able to predict tissue state. So the input, again, is the patient MRIs. We have local regional texture features. We have labeled biopsies and unlabeled samples that we know something about for logical reasons. We have their histology, and that histology breaks them into which tissue state they are. And this is kind of a clumping of what those 3 tissue states they are for training. And then we just asked, what is the MRI patterns that can predict this? And it turns out it doesn't do a very good job. It turns out when you do that, the accuracy of the model is kind of crap. But what you do find is if you pre-inform this model with knowledge about some baseline model of what the biology is. So in this situation, normal brain, where there's lots of neurons, is not going to have a lot of tumor cells in it. Or not a lot of reactive cells in it. That would be an easy, expected, like sort of baseline model. And you can get this more and more complex over time, but that's basically what this nested model we built was. We had a simple model that said, where is the normal green stuff, the diffusely invaded brain? Do a simple segmentation on that, input that into the system, and then say the only things left now are the choice between is it more inflamed, immune, or is it more proliferative state? And then by adding that layer, our accuracy was achievable. And I'll show you, I'll just explain to you just briefly that the way this was incorporated is through the loss function. Specifically, the knowledge loss here, the knowledge is effectively where we're putting in the mechanistic model here. And the mechanistic model is extremely simple that where there's lots of neurons, where there's lots of normal brain, there is not going to be also highly inflamed or highly tumor invaded. And that's consistent with all of our data. So going back to our tissue states, we apply this to a suite of different models. You can see all the suite of different models on the bottom are the ones where we're trying to build. So just read low accuracy. You don't have to read the details. But you can see the accuracy is quite low, like really low, because that's telling it we haven't informed it anything about the biology. We have not given it the mechanistic model. The accuracy shoots way up to these numbers with the green arrow in predicting the diffuse state when we give it that information, when we include this knowledge information into the learning. And that allows us to build these beautiful spatial maps of the complex ecosystem of a more proliferative tissue state from a more immune ecosystem, and then apply that to tracking patients over time. And so in summary, we know every patient is different because we're all part of a rainbow. Is a rainbow allowed here in Florida? Just checking. Thank you for bringing this here again, Sandy. We appreciate it. Every region of every tumor is different. Now we're getting into politics. I got told off for this once. It was fun. And then imaging tell us some about the heterogeneity, but what we classically think about it in terms of imaging telling us something is not that great. Building these models that glues this information together really can allow us to detect shifts in the tumor ecosystem that are clinically significant, although I didn't tell you a lot about that right here. So thank you. Probably 2, but that's it. Okay, there. I don't. Yes. You want to do it? I can.
Thank you. That was really cool. When it comes to the invasive diffuse phenotype, there clearly must be a spatial
Yeah.
bias.
Correct.
But what about the immune? And like you start to show the predictions for that at the end, but like, because of course we can, when you look at low dimensional projection, we're all guilty sometimes of thinking that's like space, but it's not. So when it comes to the immune phenotype, what's the spatial heterogeneity? Is it diffuse? Is it localized?
It's very diffuse over the region of the frank abnormality. It's quite heterogeneous, and I think that has to do with a large role of vasculature, because the T cells are really T cell enriched environments are only really perivascular. So I think there's a big game there that we're not there yet, because that's one of the things we want to do is like then look at the maps, better understand the regional predictions from the machine learning. That allows you to then build more mechanistic spatial temporal models that are like the PI model, but with immune populations that are mixed in, and better understanding how those interactions are playing out. So we have that from the spatial transcriptomics and that, but that's actually our next, that's what the U54 is about. So
very cool.
Thanks.
Hi, thank you for a wonderful talk. I'm a machine learning person, so I have some machine learning questions.
Sure.
I was curious if the archetypes that you found, the 3 archetypes in the 3 branches, whether those can naturally come out of the latent embedding in your model, or can you enforce the latent space to be like a 2 simplex and have that come out naturally rather than kind of coding that into the model itself?
That was our first attempt was to organically just come up with it. We kind of came up with 3, but I think we were a little low on the N. I think it's just the 202 samples for that, which is not quite enough to be able to ask the machine to handle all of this information, imaging information, and the limited samples. I think once we finally process this like 900 cohort, I think we'll be in a sweet spot where the organic nature of convergence on that side versus what I've told it on this side will come together.
And the second question has to do with the adversarial training, which is part of your architecture itself. We've used that to make things, pseudo-invertible or to add additional structure to the latent space. I'm just curious what that adversarial path, that GAN path is doing in your architecture.
In this situation, this is so if you have a highly proliferative state, it's also exclusionary to immune, right? So they're kind of on a teeter-totter's end. So we use the regional information that Adam was asking about the diffuse invasion to give us kind of like, we know that's the diffusely invaded cells, more chances than not, our guess is out here. And then the adversarial bit was the interaction between the proliferative versus the immune state and allowing them to sort of compete with each other. I don't know how else to describe it, but that's my layman's understanding of how to describe it. Did you have a question, Heiko? And then
I do.
We should move on to the next. Okay.
I do have a question.
Go ahead.
So I've been for a long time a fan of your PI model, and it has been really cool. And now you get the additional information that some tumors are more proliferative and others are more immune, inflamed, whatever. If you now separate your cohorts into the more proliferative phenotype, do you feel that do you know that your PI model works best on those versus the others? Can you now use this to pre-stratify?
Yeah, no, it's a great question, because the PI model just assumed only tumor cells present, right? The only population was tumor cells, and it's kind of like the point source data with your work, right? It's like you just assume it's all tumor cells, but it really is this admixture, and I don't know the answer, but I'm keen to look once we produce more of these. Thanks.
Okay, thanks again, Kristin.